Hello my dear lovely friends. Today in this video we'll study about the lavender top tube and the purple top tube uses. In clinical setting, blood samples are collected in this tube and sent for different tests to see the diagnosis or rule out the diagnosis. So it is the workhorse in the lab world because it is used most frequently, most reliable, most versatile tube in the entire tray in the lab. So the secret ingredient used in this vacutainer is EDTA, ethylene diamine tetraacetic acid, in two forms, in dipotassium form and in tripotassium form. Dipotassium is in the form of spray, dried form coated in the vacutainers, and K3 in a liquid form. It's a chelating agent and powerful anticoagulant that binds calcium ions and prevents clotting and keeps the blood in whole form, in liquid form, and preserve the shape and integrity of the RBC. So, since the calcium is a natural glue that helps in blood clotting cascade, so when EDTA binds with it, it prevents the clotting. The common test sent in this vacutainers are CBC, complete blood count. In complete blood count, we see the RBC, HB in it, WBC, platelets, and etc. Means different cells of the blood. Second, the HbA1c. In HbA1c, hemoglobin A1c, we see the glucose stuck in the hemoglobin in RBC. So, since EDTA preserves the shape and integrity of RBC, this sample is sent in EDTA vacutainer. In HbA1c, blood sugar of last 3 months is evaluated. Third one, blood film smear. The peripheral blood smear is sent in this to see the shape and size of the cell like malaria, PS vermis, sickle cell, etc. Fourth, cross match and blood grouping. Samples are sent to the blood bank in these vacutainers. Then the ESR, erythrocyte sedimentation rate. The standard vacutainer for this is the black, but some automated machines or machines can evaluate from the vacutainers also. The next one is HPLC, high performance liquid chromatography. Then the reticulocyte count. The retic cells, the baby red blood cells, are seen in this reticulocyte count test. Then malaria test, then the thalassemia. The types of thalassemia, in this the different variant of HB is seen to see the genetics one. Next, the toxicology. The chemicals or drugs in the blood are seen in the toxicology test. It depends, means different vials are preferred for this, like gray and heparin also. Depends on the doctors for what he is seeing for. Pro tips to be kept in mind whilst collecting the samples in these vacutainers is the inversion rule. Slowly, after collecting the sample, this sample should be inverted slowly 8 to 10 times. It should not be shaken, otherwise hemolysis will happen. Then the fill rule. The sample to be collected up to the demarcation or mark given in the vacutainer, because the ratio of EDTA and blood is very important. The more EDTA, if the EDTA is more, then there will be crenation. So the blood cells will get shrink and the study or morphology of the cells will not be accurate. So the decrease in the volume of MCV and platelet can be, so wrong reports can be seen. Next one is the order of draw. The sample should not be collected first because to prevent the contamination of EDTA. After gray and heparin tube green only, sample should be drawn to these vacutainers. So thank you. If any doubt, Please comment on my section.
अभी बंद मत करना.